Hey everyone, it's Dr. Marcon. This is the discussion on how to do the respiratory tracing calculations for Lab 2526. Before we get into that, just wanted to explain the apparatus that we would be encountering in lab. So basically, a volunteer would be hooked up to a respirometer. And the tracing basically is showing a two-way airflow. So a person would wear a nose clip, and then we would use the respirometer to measure air going in and out of the lungs, not through the nose. So that's why we have them put on a nose clip here. This is a closed system, so the person will be breathing air that is in the system, not introducing air from the nose. So we can see that the respirometer will be hooked up to this apparatus here. So we have this tube that's hooked up to a mouthpiece, and then the tube will then be attached to an inverted bell filled with air or oxygen. The volume of the bell and the volume of the student's respiratory tract create a closed system. So we know that oxygen will be used up over time, which increases the carbon dioxide content. And the carbon dioxide content will eventually increase respiratory rates. So part of the apparatus actually has a container filled with soda lime crystals that will remove carbon dioxide from the air. And the bell will go down. And as the bell goes down, the tracing in general will go up because we are actually removing a volume of carbon dioxide. Now, when we look at the tracings, we'll be able to see that the slope, if we see this next slide, we can see that the slope is going up. So here we can see the slope going up, meaning the body is using oxygen in the bell and replacing it with carbon dioxide. So we have tracing paper that is moving at a specific speed, and usually the question will tell you the paper speed. And then we're using this to measure inspiration and expiration over time and look at different capacities. So again, this will show long volumes. If the tracing goes up, that is due to inspiration. If the tracing goes down, it will be due to expiration. So we will be measuring long volumes. We cannot directly measure residual volume. We usually estimate it based on a portion of vital capacity. This is basically the air that's left in the lungs after expiration. So due to essence of time, the only capacities that we are going to be looking at and doing the tracings for include tidal volume as well as vital capacity. And I will be explaining how we can interpret those tracings within this discussion video. So when we look at the actual tracing paper, you'll actually be able to find this on page 146 of your lab supplement. We can see that the recording paper is actually divided into 1 millimeter squares. When we calculate volumes, we will be measuring up and down vertically on the page. So up, of course, is for inspiration, down will be for expiration. So we'll be calculating capacities, and then we'll be using certain correction factors to take into account various factors. So for example, we are going to be using what's called a Bell correction factor. The Bell correction factor adjusts for volume. So we know that 1 millimeter box is equivalent to about 30 milliliters. And all volume calculations must utilize the Bell correction factor. Another factor that we are going to use is known as the BTPS correction factor, or the body temperature pressure saturation correction factor. We're taking all of these factors into account. And then we are standardizing these using the correction factor of 1.1. So again, this accounts for the changes in body temperature, atmospheric pressure, and the saturation of gas with water. Okay, so then we will be using this formula once we are able to measure our capacities in millimeters by the number of boxes, and then plugging in all these correction factors to get the volume or the capacity. With regards to speed, time is actually measured across the page from left to right. The kymograph speed will be given in millimeters per minute, and then we will count the boxes in millimeters to determine the time. For example, if we have a speed of 50 millimeters per minute, so that means we have 50 millimeters or boxes in 1 minute. Okay, and usually the kymograph speed will be given. So we know that paper speed can be increased or decreased. With the respirometer, there is a button that can increase or decrease the kymograph speed. So normal standard is 50 millimeters or 50 boxes per minute. This can be decreased to 25, for example, if we need to measure for 30 seconds, or we can increase it. So measured values may be slightly different than the key. There will be some examples that you can do. If it is 1 or 2 boxes, that's okay. However, if it's a large number of boxes, a large variation, then perhaps you should double check the number of boxes you counted. Huge differences might be due to human error. With regards to counting the boxes, it's not so bad for tidal volume. But if we have large capacities, what's great is if you actually look at the tracing paper up close. This is actually on your page 146 of your lab supplement. So we know that one little box is equal to 1 millimeter, but we know that we have large boxes as well on the tracing paper. And there are 5 little boxes to 1 large box. So if we need to calculate large capacities, we can actually count the number of large boxes and then use the little boxes to add on to however many we counted with the large boxes. So if we, for example, if we look at one of the capacities for vital capacity, the peak is very large. So let's see if we can go all the way to that picture. Here. So if we look at this figure is actually also in your book. It's the one on the right. We can see that this capacity is quite large. So we need to measure from the lowest peak to the highest peak. This would be the vital capacity. So if you look at your paper, we can see we can count the large boxes. So we have about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25 large boxes. So 25 times 5 is about 125. And then we have like, 3 up top, and then 1 or 2 at the bottom. So 125 plus 5, so we're going to say it's about 130 millimeters. Okay, so make sure you utilize the large boxes if you need to measure a large capacity such as vital capacity. So here are just some general notes for the respiratory problems. So we know that the recording paper is divided into 1 millimeter sections. You need to measure the number of millimeters between 2 points, and then we use our correction factors to adjust for volume. So we use the Bell correction factor to adjust for volume. This factor is equal to 30 millimeters per 1 millimeter. To calculate volumes, you need to move up and down the page. Because the bell is hooked up to the writing pen via pulley, all tracings that move towards the top of the page will correspond to inspiration. So when the tracing goes up, that's inspiration, while all tracing moving towards the bottom of the tracing are expiration. We then have to adjust for the BTPS correction factor that I told you guys about. This will account for changes in body temperature, atmospheric pressure, and the saturation of gas with water. And we will estimate a standard value of 1.1. Okay, so that's the BTPS correction factor. Time moves across the page. In order to calculate time, we just look at the kymograph speed and measure the given distance. For example, if the paper is moving at 50 millimeters per minute, just count 50 millimeters, and that is 1 minute, okay? Again, your measured values may be slightly different from the instructor. You could be a couple millimeters off and still be correct. However, if it's a large number, make sure you're double checking your work and counting the number of millimeters. So the first tracing that we're going to look at is the tracing at rest. And again, this is on page 146 of your supplement, the upper left tracing. We are given, again, the paper speed. The kymograph paper speed will be 50 millimeters per minute. We have our bell correction factor at 30 milliliters per 1 millimeter. We have our BTPS correction factor of 1.1, and this is the formula that we will use for volume or for capacity, okay? So we get the number of boxes in millimeters, multiply by the bell correction factor, which is 30 milliliters per 1 millimeter, and then we put in the BTPS correction factor. Luckily, there are no units for it, so we just use the number 1.1. Once we are able to get the volume or capacity that we're looking for, in this case we're looking for the tidal volume, we should be able to find the respiratory rate. And the way we find the respiratory rate is, we count the number. You can either do peaks or valleys. I like to count the number of dots at the top, and then this is our tracing within 1 minute, so we can count how many dots there are, and that will be equal to the respiratory rate. And I'll show you guys that figure in just a little bit. So once we are able to get our tidal volume from this formula right here, we then take our respiratory rate, and then we can get the minute ventilation, or VE. We'll also be able to, just by looking at this tracing, be able to find oxygen consumption, or VO2. And I'll go over that once we get to that tracing. So we can see, again, the slope is going up, meaning the body is using oxygen in the bell, and then replacing it with carbon dioxide. Now, within the apparatus, the bell has soda lime crystals, which will remove the carbon dioxide and make it lose volume. So as the volume goes down, the bell writes higher, so meaning the volume is going down, and this slope goes up, okay? So for the first part, for rest, we are given this information, we are given the paper speed, we know the bell correction factor is 30 mLs per 1 millimeter. We will be calculating tidal volume, okay? So this is volume just breathing in, breathing out at rest, inspiration, expiration. So just the normal air that we are breathing in, breathing out. We'll then be able to calculate the respiratory rate, and then from these two numbers we'll use the formula to calculate minute ventilation, and then from the tracing we can also see oxygen consumption. So again, in order to calculate volumes and capacities, we use that formula which I showed you guys. We have our bell correction factor and then our BTPS correction factor. So here is that first tracing at rest, okay? It's that little corner, upper left-hand corner that we see on page 146 of your supplement. So we can measure from the bottom to the top to get our tidal volume. So if it's going up, that is representing our inspiration at rest, so that is tidal volume. We can count the number of squares from the bottom of this line to the top of this line, and we can roughly count about 18 boxes or 18 millimeters for tidal volume. Okay, and then we have our paper speed. So from the beginning of the tracing to the end, we have 50 millimeters equals 1 minute. So we use that value, and then we can start plugging in our numbers for the volume capacity formula. So I'll just move ahead, click ahead really quick. So we can see, using this formula again, volume or capacity equals the number of boxes times the Bell correction factor times the BTPS correction factor. And then using the numbers that we obtained by counting the boxes for tidal volume, it was 18 boxes or 18 millimeters. We then multiply by our Bell correction factor, 30 milliliters over millimeters, and then multiply by the BTPS correction factor, 1.1. We cancel our units. So the tidal volume we are calculating to be 594 milliliters per breath. Okay, so that is our tidal volume. Now for our respiratory rate, if we go back to our tracing, again we can count the number of peaks within this tracing, and we can count, it's already done for you, we count 14. So that means we have 14 breaths in 1 minute. Okay, once we have calculated or counted that, we can then again use that in a formula. So here we are going to use the formula for respiratory rate, 14 breaths per 50 mm, because we have our paper speed, and then we multiply that by our paper speed, and we get 14 breaths per minute. Okay, so we have the 50 mm cancel out, and we have 14 breaths per minute. So now that we have our respiratory rates, we can use these values, the respiratory rate as well as the tidal volume, to get minute ventilation. Okay, this is the amount of air that we are ventilating within 1 minute. So the formula for minute ventilation is we multiply tidal volume by the respiratory rate. Okay, so we've already obtained these values. We know that tidal volume is 594 milliliters per breath, and then we multiply the respiratory rate, which is 14 breaths per minute, and we get 8,316 milliliters per minute. Okay, so that is how much air a person is moving in and out of their lungs per minute, or ventilating per minute. So our last variable that we are going to get is oxygen consumption. And we can, again, use this tracing to kind of help us determine what oxygen consumption was for a person at rest. So we are actually going to use the slope of the line to help us measure oxygen consumption. So we know that it is going up because as a person is breathing into and out of the respirometer, we have, again, those soda lime crystals that are basically absorbing the carbon dioxide and taking that volume out. So we are going to draw a triangle to account for the amount of oxygen that is being consumed. Clinically, we get oxygen consumption another way, but for lab purposes, this is how we are going to measure oxygen consumption. So we draw a line from where we started our tracing at the bottom to the end of where we stopped our tracing. And again, we have our paper speed at 50 millimeters per minute. And then we can see that within 1 minute, we see this change here of about 17 millimeters or 17 boxes, and this is what is going to be used to measure oxygen consumption. So we have to assume we are using some of the oxygen in our normal metabolic process while hooked up to this respirator. And we replace it with carbon dioxide. So we're going to assume that within this apparatus, there's a 1-to-1 ratio of oxygen being consumed and carbon dioxide being produced. So for every carbon dioxide that is produced, the lime crystals in the apparatus will actually remove it and decrease the volume, which is why we have an increase in the slope of the tracing. Okay, so we're going to draw a triangle from the beginning of the tracing to the end, and then count the number of boxes that the tracing, that the slope has gone up. So this shows you how much the bell has actually dropped because that volume is being taken away due to the lime crystals. And again, this will give us the oxygen consumption. And we counted that to be about 17 boxes or 17 millimeters. So the bell has dropped 17 millimeters in 1 minute. Okay, so now that we have counted that, we will take that 17 millimeters, we'll then again multiply it by the Bell correction factor of 30 milliliters per minute or per millimeter, and then multiply that by the BTPS correction factor. So 17 millimeters times the Bell correction factor of 30 milliliters per millimeter, and then the BTPS correction factor of 1.1, and we are going to get oxygen consumption at 561 milliliters. Okay, so now we are able to determine all these values from that top tracing, and this is the tracing at rest. Now below that tracing is the tracing after doing exercise, and this is something for you guys to practice just so you're comfortable doing these respiratory tracing problems. And the next couple of slides actually goes through step by step the tracing of the figure at rest that I have gone over with you, so you can reread that and get a better sense of what you're supposed to do. But basically everything I just explained to you are in these next slides. How to get tidal volume, and then plug it into the formula. Then how to get respiratory rate. And then your minute ventilation, and the formula that you can do after you've gotten tidal volume and respiratory rates. Okay, and then oxygen consumption. Again, this basically writes it out for you, how to get the oxygen consumption and the theory behind that. And then here's that formula, putting in the Bell correction factor as well as the BTPS. Okay, so below that tracing, you have that tracing for exercise. I'm not going to go through it, but know that it's basically the same steps as the tracing for being at rest. What we can see, we can already see there's a huge difference between this tracing and the rest tracing. We can see that the lines are a bit longer, meaning more oxygen is being consumed, and more carbon dioxide is being blown out. And we can see that the tracings are a lot closer together, so the respiratory rate looks like it's a little bit faster. So we can definitely see that there is a huge difference between the tracing for exercise and the tracing for rest, and you guys can actually, well, now that you see the steps for rest, you can do the steps for the tracing for exercise. So, and again, it actually explains to you step by step what you need to do. So, here are our correction factors that we need to take into consideration. We have our paper speed. So again, you will be asked to first get the tidal volume, then get the respiratory rate, and with these two factors we multiply together to get the minute ventilation, and then going back to the tracing we can then determine oxygen consumption. Okay? And here are the formulas kind of laid out for you. So again, exercise we can observe the capacities are closer together, the slope is higher, so meaning we are consuming more oxygen. And again, we would perform all those steps like the steps that we got for rest. We perform the steps for exercise and get those values. Okay. So the next thing that I do want to explain to you is vital capacity. So actually here is a nice table showing you the differences between the two tracings. So the one at rest here, the one for exercise, and we can compare the tidal volumes for rest versus exercise. And we can see that all variables do increase with exercise. So tidal volume increases, respiratory rate will increase, minute ventilation will increase, and then oxygen consumption, of course, will also increase. Okay, and then just another table showing you the comparison of the two. So the next figure, which is also on that page 146 of your supplement, we are going to be taking a look at vital capacity and tidal volume in this tracing. So the student that's hooked up to the respirometer will take a maximum, after breathing, a few times normally, the student will take a maximum breath in and then blow it all out. And we can see the line going up for inspiration and then down for expiration. Okay, and we actually we used this tracing earlier to show that you can use big boxes to help you count the number of millimeters from the bottom of the capacity to the peak. And then we place that into, again, this volume capacity formula that we used earlier. So we have already counted the number of boxes earlier. We counted 130 millimeters from the bottom of the peak to the top. And then, again, placing the Bell correction factor of 30 milliliters per 1 millimeter, and then the BTPS correction factor of 1.1. Okay, so again, we're only taking a look at tidal volume and vital capacity. For testing purposes, these are the only two that we're going to be concerned about. You don't have to concern yourselves with the other capacities such as inspiratory capacity or expiratory reserve volume or functional residual capacity or total lung capacity. So we're only looking at tidal volume and vital capacity. Okay, so you will not need to know that for the exam. So again, here we see that figure and compare it to the other capacities. Again, the next couple of slides will give you step-by-step instructions on how to get tidal volume and then how to get the vital capacity. So for tidal volume, again, we measure from the bottom of the tracing to the top, and we can see that we get about 15 millimeters for tidal volume. And then we plug that into the volume capacity formula, accounting for the Bell correction factor and BTPS. So here, tidal volume, we get to be about, we calculate to 495 milliliters per breath. Okay. And then for vital capacity, let's see here. Again, we're measuring from the bottom to the top, and we measured it out to be 130, maybe 129, and again, putting in that for the formula, volume capacity formula, putting in the Bell correction factor and BTPS correction factor. And here, vital capacity, we find at 4,257 milliliters per breath. So that's it for this discussion on respiratory physiology for this lab. Again, you'll need to be able to look at a tracing for rest versus exercise, and know that with exercise all variables will go up. So you need to be able to count the number of squares to get tidal volume, and then be able to get the respiratory rate. And then once you have tidal volume and respiratory rate, you'll be able to get minute ventilation, and also you can take a look at the tracing to get oxygen consumption. For the third tracing that has that large spike that we can see, this is for vital capacity. You just need to be able to get tidal volume as well as vital capacity for this tracing, and that is what you're going to be tested on for the lab exam.